Okay, I've got 1:15, and I know we just had lab, but is there anything pertaining to lab you'd like to talk about?
Can I ask about the exam?
Yeah.
A
Sure.
question about the exam. The pin on the back of the knee. Can you?
Uh-huh.
Are you allowed to tell me the answer?
I think I put anterior cruciate.
I put posterior, so that's why. Was it anterior? I put posterior.
Oh, where's my answer key? I don't have it.
I thought it was posterior.
I'd have to go in and look, to be honest with you. I just cannot remember. I
Okay.
still have the station still set up, so I will check when I go in after this lecture. I had the meeting just until a second ago, so.
Gotcha. I was just curious because it threw me off a little bit, but
If it was in the back, I'm assuming it's probably posterior. That's more than
Okay.
likely what it was.
Okay.
But I will go and double, triple check. Anything else with regard to lab or what's coming up in lab or anything like that?
I just have another question about the lab practical today, like one of
Yeah.
the stations. I don't know if you'll remember it. It was station 17. It was like, where in the body would you find this connective tissue?
Yeah.
So
go ahead.
was it hyaline cartilage?
No, it was elastic.
Oh, it was elastic.
I'm pretty sure it was elastic.
Okay, so you could have put like ear.
Yeah.
Okay.
Epiglottis, ear. Because you could see the dark stained fibers. That was what should have triggered elastic versus hyaline. Now another station did ask what kind of tissue is being shown, and that was hyaline cartilage. I don't recall what station it was, but it was one of the middle stations, I think. Yeah. Anyway, so I'll take a look at those this weekend or tomorrow or sometime. Get them back to you next week. Anything else on lab stuff? So in lecture, as I said, in lab today, we have the muscular system on the docket, I believe, on the schedule. This is our 10th week already, which means next week we start respiratory system. So we've had about a week and a half. By the end of this week, it will be a week and a half according to the schedule on muscular system. And this particular chapter does require some study, in particular the microanatomy, if you will, of the muscle fiber. You know, knowing what the thick and thin filaments are, how they overlap one another, knowing what a sarcomere is, understanding how myosin heads grab onto the actin filament during contraction and cause the Z lines to get closer together, et cetera, et cetera, et cetera. So it's fascinating, interesting stuff, no question. Of course, what isn't? This is all interesting.
So all like the I bands and the Z lines and the A bands and stuff, like all that we should know. And so, okay. And the myofibrils, basically like their sarcomeres joined, right?
The sarcomeres butt up against one another, right, as the Z lines. Those are what separate the individual sarcomeres.
Okay.
Yeah. So I'm going to pull up chapter 9.
Those are myofibrils, like end to end, though, right? That's what it says on 2696. I just was sort of.
Let me pull up. Let me pull up the PowerPoint.
Sorry.
Chapter 9. Okay. So this slide sort of summarizes the preceding 3 or 4 slides in the PowerPoint. And again, absolutely critical that you understand this slide. Really, really important. So when we talk about the sarcomere, which is the structural functional unit of skeletal muscle, here we're seeing one of the myofibrils that lie within the larger muscle fiber being pulled out, right? And we can see the thick filaments here in the center of the sarcomere. These are the pink guys. And then we have the purple. Those are the thinner actin myofilaments. So these filaments, the thick and thin myofilaments, fall within or lie within the myofibril or fibril. And then there are many of those in each muscle fiber. And these sarcomeres, again, butt up against one another. As you can see, you know, here's the end of the preceding sarcomere, here's the next one, and then here's the beginning of the third one, with no Z line visible yet on this particular sketch. So there's Z lines all along the length of the myofibrils. So when you look at skeletal muscle like we did recently in lab, and you go under high power and you see the dark light striations, right? This is a much more magnified view of this, but here's the light, here's the dark, here's the light. Can you picture what I'm describing? What you're seeing here in terms of light, if you will, light striation, this is actin. Where you see dark, this is myosin and some overlapping of actin. And now we see, again, more solely actin. So if you can transition, you know, from this diagram to this diagram to this diagram, then you can understand the anatomy. But it requires study and review.
So the sarcoplasmic reticulum, is that, it says it responds to the endoplasmic reticulum. Is that like it's not liquid, it's like a coating over that?
The sarcoplasmic reticulum is this organelle shown in yellow.
Yeah.
And you can see it in between all of the myofibrils here within the muscle fiber. Now, of course, these are cross sections that you're seeing up here, but this is mitochondrion in orange. And then these little yellow tubular structures are all part of an interconnected sarcoplasmic reticulum. And its function is to store calcium. That's what its primary function is, storage of calcium. So when a muscle impulse comes down the sarcolemma or cell membrane and then enters the transverse tubules, and again, I'm not sure if you've studied this or this is the first time you've heard this. Let me just pause here and ask, how many people have not studied this yet? And you can be honest with me. I'm not going to yell at you too much, just a little bit. What?
I just kind of read it, basically. I didn't really study, you know?
Yeah.
Yeah, no, that's,
the above.
yeah, I get that. Make a goal of getting into this though, you know, soon. Because if I start talking about this, which I'm happy to do, it's going to make a heck of a lot more sense if you've looked at this before. Otherwise, it'll be a little confusing. And even if you've looked at it before, it can be a little confusing. So let me lay out the process a little bit. You know, today in lab, we talked about the neuromuscular junction and the muscle model, right?
Yes.
So that's what this slide begins to talk about. Here's the axon of the motor neuron.
Oh, I can't see it.
Yeah, we can't see your screen.
Oh, okay, sorry. Thank you for saying something. Okay, now. So this is the diagram that references the motor neuron coming down here. Here's the tip of the axon. And it makes very close contact with the sarcolemma of the muscle fiber, which is the cell membrane of the muscle fiber. It's called the sarcolemma. This is called the motor end plate of the sarcolemma. See how it's very folded here? Any idea why it's highly folded?
And
Well,
for more
anytime-
surface.
Increases surface area, exactly. That's the answer. Whenever you're asked, why is this folded? The answer is always increased surface area.
Throw
Well,
that out there.
yeah, but the question now is, why do we want increased surface area? Well, if you've not read this or studied this, you don't know the answer to that. Does anybody know the answer to that? Why is this sarcolemma so highly folded at the motor end plate near where the axon comes down? And by the way, they don't actually touch. They get real close, but they don't touch.
I mean, just more area to feel this, like to push the impulse through. I mean, I'm just guessing.
Well, you're real warm. In order to take a nerve impulse and convert it into a muscle impulse, resulting in contraction, you've got to have receptors here in the sarcolemma that will combine with the neurotransmitters released from the synaptic vesicles here of the axon. And again, this is the first time you've heard of this, a lot of you, so this is all new. You need to get into chapter 9. So these blue structures here, these dots, represent a particular neurotransmitter called acetylcholine. That's the name, acetylcholine. Now, you've heard of neurotransmitters. If you've taken psychology, you learned about serotonin and dopamine and that kind of thing. Those are neurotransmitters. There's a whole slug of them. This particular neurotransmitter is being released from the axon, shown in yellow, via these synaptic vesicles. They're organelles that lie in the axon. And so they are dumping their neurotransmitter acetylcholine across this cleft, this space, in response to a nerve impulse. And then that acetylcholine will combine with receptor sites here on the folded sarcolemma. So by having the folded sarcolemma and increasing the surface area, you can pack more acetylcholine receptors. That's why. Once that happens, and there's a video that I have placed here, a hyperlink. There's a video here you should watch. Actually, maybe, well, there's 2 videos, but this is the one that talks about the neuromuscular junction. Once that neurotransmitter gets released and combines with receptors, then what happens is we have what's referred to as depolarization of the sarcolemma. That's a fancy way of saying we generate a muscle impulse. So we go from nerve impulse to muscle impulse in response to the release of the acetylcholine across this cleft. And then that muscle impulse is going to travel down the cell membrane, the sarcolemma of the muscle fiber. And those muscle impulses will go into these little holes. They're called transverse tubules. See all these little holes? And once that impulse goes inside, and here we are inside the muscle fiber now, in between the myofibrils, you can see that these T tubules, called transverse tubules, because they run transversely at a right angle to the length of the fiber, that causes the sarcoplasmic reticulum, shown here in yellow, to release its calcium. And I have a little box here that talks about that very process. And it's the release of the calcium that's so critically important in terms of causing interaction of the myosin thick filament and the thin actin filament. That's what causes contraction. So you really need to get into the chapter and watch the videos, read the book, and then you can formulate some understanding of the process. And it's really very interesting how it works. And again, I've got videos embedded within these PowerPoints, so there's plenty of good stuff to watch. But this is a fairly long chapter. Whoops, what happened there? So you guys really haven't got any questions because you haven't gotten into the chapter yet. Is that basically the case? Yeah. Well, maybe we should just start at the beginning of the PowerPoint, just work our way through, see how far we get, you know? Why don't we do that? Okay. So this scanning electron micrograph, does everybody see it?
Yeah.
Okay. Here we've peeled away a little bit of the sarcolemma, and we can see the myofibrils inside the muscle fiber. There's hundreds of them, really. So this big whole thing, this is one muscle fiber. Yeah. Magnified 3,000 times.
I feel like everything in the muscle system is so nervous related too. It's like we're so wrapped up in the nervous system as well.
Well, it certainly responds.
I mean, I just never thought of it like that.
You know, it responds to the nervous system, right? So they are intimately tied together, you know, via the neuromuscular junction, for example. This is a view of 4 fibers, and reminiscent of what you would see if you looked at a slide in our lab, right? And you can see the dark and light striations, like I talked about earlier. And we can also notice the many nuclei here, because remember, skeletal muscle fibers are multinucleated. And so the next few slides kind of talk a little bit about some connective tissue that helps to bind muscle to bone. We've learned about these before, right? The dense, regular connective tissue that make up tendons and ligaments if it's bone to bone. We also have what are called aponeuroses that often help to bind muscle to muscle. And the best place to look for an aponeurosis is here in the abdominal region, between the rectus abdominis muscles. Well, actually it's one muscle, but the components of the rectus abdominis you can kind of see here. You know, the six-pack ab look when you do a bunch of crunches. It takes a lot of crunches to get abs that look like a washboard, but what connects those parts of the rectus abdominis muscles are pieces of connective tissue, broad sheets called aponeuroses. So it's just kind of interesting. It's muscle to muscle connection. You might also recall in lab, we identified the frontalis and occipitalis and temporalis muscles. Remember that this morning? Well, those are also connected to the skull bones via these aponeuroses as well. They're thin sheets. That's what they are, thin sheets of dense fibrous connective tissue. So they're histologically similar to a tendon or ligament, only they're broad, thin sheets.
So I have a question about those. So if you were to get like an abdominal surgery, is that going to be difficult to heal because of like, is it thin enough where it has a good blood supply, unlike, the kind of bigger like
Yeah.
bands of tendons?
Right, I understand your question. I don't know the answer, to be perfectly honest. I can hypothesize, but it's just a guess.
Yeah. Okay.
And my guess would be that it would take a long time to repair. Just given the fact that dense regular is typically not very vascular. However, do not quote me. Because it's close to muscle, maybe that's a different story. I just don't know. Well, you tell me. I mean, maybe you don't know, but if you've had a C-section.
Well, that's what I was thinking. I work with a lot of new moms, and a lot of them say, "Oh, it's a year later, and I still feel like my stomach isn't,
Yeah.
healed all the way or whatever."
Yeah, sure. Well, one thing they're going to certainly do when they make that incision is they're going to cut down the length of the aponeurosis and not across it, right? So by cutting down the length of it is certainly going to be a better scenario than if you cut across it, I would think. I think that's probably a fair statement to make. I'm not a surgeon, but I think that stands to reason. And again, I don't know the procedure very well, but I'm assuming they're probably trying to avoid cutting through the muscle itself. They can go right down in between the muscles, like here. Although this is not the only abdominal muscle, I have to tell you. There's actually a thin layer of 4 muscles here in the abdominal region. This is just one of them, the rectus abdominis. There's transversus abdominis. We're going to learn about these next week. There's external oblique and internal oblique. There's 4 layers. They're super thin, but when you do a C-section, you're cutting through all 4 layers, plus some connective tissue here too. Yeah. But that's a really good question, Paige, and I don't know to what extent. I mean, having a baby, that in and of itself changes the physiology and the anatomy of a woman. It just does. And so respective of that whole event and its long-term impact, I'm not exactly sure if it takes a longer time to maintain that or get that tone back. If let's say she was a fitness model or really was working out a lot and she took her post pregnancy seriously, whether she could get back to that same tone in as quick a time as if she had it vaginally, I don't know. But great question.
I know that like if they do like an emergency C-section, it's different, but if it's planned, they do like they cut it better. I know that yeah, it's weird. But like it literally cuts the muscle wall. Like my sister literally has like a shelf and she's not heavy or anything and she's worked out and it just you can see like just that it it was separated. Totally.
Interesting. So here we're now beginning to work our way into skeletal muscle. Now this is obviously showing the femur. You see the greater trochanter here in the head of the femur. But this could be any long bone. We could even pretend this is the bicep or triceps, coming off of the humerus, for example. We all, I think, know where those are. But we know that tendons connect muscle to bone. And so coming off the periosteum of the muscle would be this very dense, fibrous connective tissue, which would come to know and love, right? Good old tendons. And then as we proceed further out, we may encounter what's called fascia. And this also is dense connective, but it can delve into the muscle and separate it. If you've ever processed a deer or a cow or a pig, then you kind of have encountered some of this connective tissue between muscle groups. If you've never done this, you have no idea what I'm talking about. But how many people have seen a deer gotten processed, or maybe you participated in that? It's quite interesting. It really is cool. Yeah, so some of you know what I'm talking about. So some of this fascia can be this kind of snotty, slippery, kind of a gelatinous, snotty is the best way to think of it. It's kind of clear, stretchy stuff. that's fascia. But you can have thicker bundles in between muscle groups as well. So let me go back to my screen
So
share.
when you like pulling like the skin off a deer and there's all that white stuff and you know
Yeah.
makes that noise, is that what you're talking
Yeah.
about?
Yeah, that's fascia. Yeah, exactly. Yep. Yep.
Okay.
Yep. Just under the fat,
Okay.
just above the muscle. That'd be superficial fascia. Yeah. Okay, and then as we get sort of into the muscle, and we've cross-sectioned it here, you can kind of see we've cut it. We have 3 additional special types of connective tissue. The most superficial of which is shown here in red, called the epimysium. Epimysium, and I'm taking my pointer here, my arrow, and I'm circling it. There's the epimysium. It's on the very, very outer edge of the entire muscle group. It's a connective tissue. So there's epimysium. And then as we go into the muscle, we see it's divided into groups of skeletal muscle fibers called fascicles. So here's a fascicle, here's a fascicle, here's a fascicle. You can see there's like what, 7?
Can't
of
really
them?
see your arrow for some reason.
Do you see the bone tendon slide?
Yep. It's just not the arrow for some reason.
Oh, you don't see the arrow. Okay, let me, can I draw on this? Let me see if I can draw on this. Can you see my red draw? Oh, what the heck is going on there? Oh, this is weird.
We're starting to see a little of it.
On the bone.
Yes.
Yeah, it should be on the muscle. Oh, this is so bizarre. Why is this doing this? I don't, there's something weird going on here. Let me try something. I'm gonna get out of this. Hang on 1 second. I want to be able to draw on this diagram for you. Try this again. What do you see?
The slide's actually in a different screen now, kind of.
Yeah, but I can see your cursor moving
You
though.
can see your cursor now. Yeah.
Okay. So let me try to just go right to the slide. Do you see my cursor now?
Yes.
Okay, great. So here's the epimysium that I was telling you about. Do you see my arrow moving?
Yes.
Okay, great. So this is the outermost of the 3 connective tissues we're going to talk about. They're outlined here in red and blue and green. So this is epimysium. And then as we go inside the larger muscle, we have bundles of muscle fibers called fascicles. So here's a fascicle, here's a fascicle, here's one, here's one, here's one. You can see there's like 7 of them, I think, on this diagram. Okay, and in between the fascicles, where I'm moving my cursor, this is called the perimysium. So outer epi, and then we've got perimysium in between the fascicles. Okay. What else do we see here in the perimysium area? What are these red and blue structures? Any idea what those might be? They're cross sections of blood
Well,
vessels.
there's the sarcomeres in there too.
No.
I mean,
No,
here.
not yet.
Not yet. Okay.
These are blood vessels.
Okay.
The red's depicting an artery and blue is depicting a vein. These are cross sections. They run up and down and through the muscle. The blood does. Why skeletal muscle looks red, dark red usually, right? Think of when you go buy a roast at the store. Hamburger, dark. It's vascular. Okay. Now, if we go inside a fascicle, in between the muscle fibers, we'll encounter the endomysium. So let me go to the next slide. We're going to zoom in here. Here is one fascicle that I've pulled out of this larger muscle. And we can see the endomysium here in gray. Note again that this is the connective tissue that surrounds the individual muscle fibers within the larger fascicle. Note that just like we had blood vessels between the fascicles, like we have here, we also have blood vessels within the fascicle, like we have here. Blue denoting veins, red denoting arteries. In fact, there's even nerve fibers coming down. Here we're looking at an axon of a motor neuron, which shouldn't surprise us because we talked about that a couple minutes ago. When an action potential or nerve impulse comes down this axon, it's going to cause, perhaps, the muscle fibers here inside this fascicle to contract. So, again, here's a fascicle defined as a bundle of skeletal muscle fibers. How many muscle fibers do we have in this fascicle? We have 1, 2, 3, 4, 5, 6, 7, 8. Looks like there's 9 of them. Now you might say, well, what are these little bitty structures I'm seeing inside each muscle fiber? Well, those are the myofibrils. So again, you're learning the microanatomy here. And at first, it's a little confusing. You got to really study it. We're going to take one muscle fiber from this fascicle, and we're going to pull it out. And there we've done that. And we can see the myofibrils inside. There's probably how many? 3, 6, 9, 12, 15 or 16 myofibrils in this one muscle fiber. We can also see the sarcolemma here on the surface. This is the cell membrane. That's all that means. Sarcolemma is cell membrane. We can see the sarcoplasmic reticulum here in yellow. It's not just here on the very outer edge of these myofibrils, but it extends in between them as well. This doesn't show it very well, but they do. That one slide I showed you earlier did depict that very nicely.
The sarcoplasmic reticulum goes between them?
Yep.
Okay.
It innervates the entire fiber in between all of the myofibrils.
Okay,
Because-
so I feel like just as like this is, you know, relatively new,
Yeah.
the fibers and myofibrils is throwing me off. So
Okay.
it is essentially a fascicle, and then which is a group of muscle cells or fibers, and then each muscle fiber is made up of myofibrils.
Correct.
Okay.
Absolutely right. Yep. And then to confuse you one last time, Paige, look what's in each myofibril. We have the thick and thin filaments, myosin and actin. Yeah.
So the video, we did do the video, then the book.
The video, yes, the video will talk about the various levels of organization. Yes.
Yeah, it makes it easier when I go to read the book after I watch the video first.
Yeah. But now maybe you can begin to see, as we talked about earlier, see the Z lines are called here in dark blue. And we can see in between the Z lines, this is the sarcomere, 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 sarcomere. So now we can begin to understand the light, dark striations that we saw earlier. They again refer to the interaction of the myofilaments inside each myofibril, inside each muscle fiber. That's why I said earlier, it's really important to get a good firm foundation of the anatomy, the microanatomy of skeletal muscle before you start to talk about or learn how contraction works or what contraction is, because it won't make any sense to you unless you understand the construction of the fiber and what lies within the fiber. So this is the slide I mentioned earlier, is a conglomeration of at least 3 slides, 3 or 4 previous slides. And what I did was I took the diagram in your book on page 295, which is this slide. And I thought, this is too darn much to give the students. There's too much information here. I would rather start them off and work through, you know, in ever increasing magnification. I don't know. You tell me if that's a good approach to take or not. I hope it is. Not that you couldn't look at this and get the same thing out of it. You could. It's just there's a lot of stuff here. This little flow chart, you know, is also very nice to reference. Where we go from broadest, largest to smallest structures. That's a real good flow chart to use. So here is a stained section through skeletal muscle. And this whole thing, do you see my pointer, my arrow? Yeah. Okay. So I'm going to trace the outer edge of this fascicle. Comes around, and then it comes way over here and comes up and around. So this is going to be the paramecium on the very outer edge of the fascicle. Okay. These are individual muscle fibers. So that makes this the endomysium, the connective tissue between the muscle fibers. And we can see the nuclei here of the muscle fibers nicely as well in purple. And then what are all these little bitty dots you see inside here? All those hundreds, there's hundreds of those dots. Those are all myofibrils.
Myofibril.
Yep. Now, in the earlier example, we just talked about 15 or 20, right? 15, 16. The reality is in our muscle fibers, our skeletal muscle fibers, there's not 15 or 16 myofibrils. There's probably hundreds of myofibrils, as evidenced by this more accurate histologic stained section. I'll just stop here and ask if there are questions. Everybody good? Okay. So now when we look at this diagram of a single muscle fiber, we can see one of those many hundreds of myofibrils being pulled out of it. Now, again, they're only showing a dozen. We know there's many more than that, but as long as you understand the concept. Here's the sarcoplasmic reticulum in yellow in between all of these myofibrils. These are mitochondria. Why would you want a lot of mitochondria here? Well, I hope you can answer that. Because contraction takes energy, doesn't it? In the form of ATP. You need ATP. And then if you're wondering, what is this lighter yellow structure here? This is part of the T-tubule system. You might remember those openings are on the surface of the sarcolemma, and we said earlier that the muscle impulse travels inside the T-tubules. And what does the sarcoplasmic reticulum do in response? Releases calcium. And that is what causes muscle contraction. So we can see the thick myofilament here called myosin in dark pink. We can see the thin actin myofilament in purple. and we can see these Z lines, and I think they're aptly named because if you look closely, they make like a zigzag, don't they? Zigzag, zigzag. That's how you remember that, Z. The material between the Z lines is the sarcomere. It's not a sacromere. There ain't no such word as sacromere. So don't write down sacromere or say sacromere. There ain't no such thing. It's sarcomere. Just like there's a sarcoplasmic reticulum or a sarcolemma. So sarco refers to muscle, skeletal muscle. And you see that prefix obviously here in 3 words, right? So a lot of sarcos. Sarco, sarco, sarco. And within each sarcomere, and I think, you know, Freya had been looking at this because she was throwing out some terms like I band, A band, and H zone and such. So you've looked at this a little bit, I think, haven't you? This gets at specific structures that we can see if we look at muscle under extremely high magnification. And so here we see a Z line. Here we see a Z line. On either side of the Z line, we have what's called the I band. Now, what is the I band? Well, let's go back. Where did I have the diagram? Okay, hang on a second. Okay. Can I draw on this? I'll try this again. Do you see my red line? No, it's not. Oh, it's drawing on the left again. Why is it doing that? Okay, I'm not going to be able to color on this. I don't know why. Do you see the slide still? Okay, so back to this lowered bit of information and then back up to the myofibril. I want you to understand, if you tell me if you can connect the information down here to the information up above. So on either side of the Z line is the I band. Can you see it up here? It goes from here to there, right? It goes between these darker myosin areas. So this is all actin, isn't it? It's all purple lines, right? On either side of the Z line. That's this region here.
So the I band goes across into like the M line and the H zone.
The I band is part of really 2 sarcomeres. The end of one and the beginning of the next. Right. Okay. Everybody follow me? Okay, you tell me, what is the H zone made up of? Can you figure that out? H zone, it's right here. Here's the one here, H zone here. There's an M line in the middle of it. You don't
Is
see
that
it.
just myosin without the actin?
Right, exactly. Perfect. It's just myosin right here. Right here. Just myosin. Well, what's this darker part of the A band, on the very outer edges of the A band?
Is that where there is actin inside there?
Well, give me a complete definition or description.
Yeah, I guess I'm not ready. Yeah.
No, I'm not saying you're wrong, but explain it to your grandfather, granddaughter, whatever. The darker region here within the A band, outside of the H zone, is what up here?
Oh, it would be where there's like those pink dots down the line, right? To the left of your cursor?
No.
No.
You're not understanding what I'm saying. I'm done with H zone.
Right.
I'm into the darker part here. Where is that up here? It's not down the middle of this. That's H zone.
Okay. So is it where the pink and purple lines cross each other?
Right. It's where they overlap.
Okay.
It's where the actin and the myosin overlap. That gives us the darkest of the dark. Do you see that? So this is this. This is this. This
Oh, okay.
is this.
So it's
You
like the pink and purple combined and make a darker color as far as the diagram goes.
Exactly. It makes perfect sense logically. Just think about
Right.
it.
Okay.
You know that actin is thinner than myosin. You should know that. Thin filaments equals actin. Thick filaments equals myosin. So if you have just thin, it's going to look light. If you have just myosin, you're going to have a darker color. If you have both, it's going to be darker yet. And that's what you've got on either side of the H zone within the A band. Does that make sense?
Yes.
And this next slide, kind of reviews what I just talked about. So if you go back and forth between these two slides, I think that'll be very clear. Again, you got to study it. It's not going to come to you the first time. You got to study it. So here's your first quiz question. What is shown here? This entire structure is what?
Muscle, like like a muscle fiber.
No,
Or
no, look closely.
like several fascicles
No, no.
or myofibrils.
Is it a
I'm just
sarcomere?
That's a sarcomere.
broke all the way down. Okay, so that's like totally magnified.
Yeah. Oh, yeah.
Oh, 16,000.
Yeah. Yeah. It zoomed in 16,000 times.
Okay.
So what are those?
The Z lines.
Right. Z lines. What are those? Just actin.
Wait, where's your cursor now? I don't see it.
You don't see it says, what are these?
Yeah, I see that, but I just don't see your cursor. Oh,
Well,
okay.
you don't need a cursor.
Okay.
I'm asking you, what are those two areas that are shown?
I bands.
Those are the I bands, right? Yep. Are you starting to see how this works? Now, this thing right here, you see my arrow? You know what this is?
And sarcoplasmic reticulum.
No. Well, you just told me these are the I bands.
Is that the A band?
The A band is from here to here.
Okay.
I'm asking you this. In the very center of the A band is the M line. This is the M line. I'll go back to right here. Here's the M line down the middle here of the A band, the very center of the myosin. Anyway, you got to study it. Okay. So there's some nice animations you should watch. Here is a review of what we just spent the last 15-20 minutes talking about. Very nice. Okay, we're going to now zoom in on an area of the sarcomere where we have both thick myosin and thin actin filaments. So what I've just circled here or ovaled here and colored in green, we're going to zoom in on them. There we go. We've zoomed in. Here's the thick myosin in purple, or pink rather, and here's the thin actin purple. Now you have to understand that this is a three-dimensional structure. A muscle fiber, a muscle cell, a myofibril. It's three-dimensional. So we would have another actin filament right here where I'm drawing with my arrow in front of these little paddles. We'd have another one over here in front of these, on top of these paddles. There'd be one in the back as well. You got to think three-dimensional, with the very, very center of that being myosin. For simplicity's sake, we're just going to show them above and below. But I think this sketch does give you a distinct impression that these myosin heads are not just above and below, but they stick out toward you and they stick out away from you on the other side. Do you understand what I'm saying?
Yeah, like it's round.
Are you all awake? Yeah, sure. You gotta think 3D. Okay. Do you see the myosin molecule in the middle? Yeah. Okay. So again,
And
go
what
ahead.
what is the purpose of the sarcomeres? Like, what is what do they do?
Well, we're starting to talk about that now.
Okay, look at that.
We have just been focusing on the structural, sarcomere. We're going to now talk about the functional aspect of it. Yeah. So you see where the thin filament is defined here as being actin, and that the actin itself is composed of this double-stranded protein wrapped up sort of like a double helix. This is not DNA, but it is a double helical protein made up of a couple different structures. One of those structures is called troponin, and one of these parts of actin is called tropomyosin. And I've shown you here, I've circled this 3 protein subunit. There's a troponin molecule. Here's a troponin molecule. Here's one. Here's one. They're all over the place. See them every once in a while? Troponin. And then we have this tropomyosin, which is depicted as this gray-blue kind of filament here on the surface of this double helical actin. Now this is still part of actin, but it's on the very outer groove here of the actin helix. So here you can see tropomyosin up here. Here's some more here. Okay. And these 2 proteins make up what's called the troponin tropomyosin complex. You'll see how they work together in just a minute. But you'll be hearing a lot about them in just a moment. You should know what they are. So troponin, 3 protein subunits scattered throughout the surface of the actin, and the tropomyosin, these kind of rod-shaped structures on the very outer groove of the actin helix. These are all proteins. Now, if you look super close at the double helical actin, these circular kind of light purple balls, do you see on the very, very edge here near where the tropomyosin is, it looks a little pink? See those little bitty pink zones? Do you see them? They're covered. They're blocked. by the tropomyosin. That is where these myosin heads are going to go up and make contact with the actin, right where those pink zones are. But it can't now because tropomyosin is blocking those heads of the myosin from grabbing onto the actin. So there's no interaction going on now. Okay. So this brings us back to discussion of the neuromuscular junction and what happens there. We talked about that earlier, right? So we have release of the calcium by the sarcoplasmic reticulum once a muscle impulse is generated at the neuromuscular junction. Remember, we talked about that. The muscle impulse comes down the sarcolemma, goes into the T tubules, and it causes the sarcoplasmic reticula to release calcium. That calcium that's released is going to combine with the troponin 3 subunit protein. And here it has done that. Look super close. Here's calcium. Here's calcium. There's calcium. They bind to the troponin, to that middle protein subunit. There's 3 proteins here, but the middle one, the biggest, is where that calcium binds. That causes tropomyosin to be displaced, moves. And look what is now available. The myosin binding sites. See them here? The pink guys that were closed up here, blocked. Now they're open. And myosin and its heads, these paddle-like structures called the head portions of the myosin protein, they can come up and grab onto the actin. And they've done that right here because the binding sites are available. Now, if you look on the myosin head, it says ADP plus P. That came as a result of ATP having been broken down. You might remember when ATP is degraded, you make ADP and phosphate. Yeah. So this is a result of a previous ATP molecule that was broken down right here on this head portion of the myosin, and another one here. In fact, all of these head portions of the myosin have ADP and phosphate there because of the breakdown of the ATP. When those constituents leave, and they've done that here, that causes the myosin head or cross bridge to make what's called a power stroke. Kind of like when you're rowing a boat. Row, row, you're moving the oars and your boat's moving. Well, here the oars are tugging against the actin, and the actin is getting pulled to the left, as the arrow shows. That happens, that power stroke, or those power strokes, because there's many of them, occurs when the ADP and the phosphate dissociate from the myosin. Almost as quickly as the ADP and phosphate leave, look what comes aboard. More ATP supplied by the mitochondria. That causes the cross bridge to detach from the actin. So here they were connected, now they detach. The ATP is broken down into ADP and phosphate. That causes these cross bridges or little head regions to recock. And then they can go back up and grab on again. And when the ADP and phosphate leave, we do a power stroke. more ATP comes on board, we detach, we break that ATP down, we recock, and we can continue to do that again and again and again. The result is that the Z lines get closer together as the actin gets pulled to the right and to the left. So here we've got sort of the relaxed state before any sort of interaction occurred. And then as we go down, here's contraction, here's full contraction. We can see that in the actual micrographs here. See how the Z lines are further apart and they get closer together as this sliding filament model works. It's called a sliding filament model or sliding filament theory of muscle contraction. And again, there's a video you want to watch that? You can watch that.
In a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins slide toward the center of each myosin myofilament. As a result,
What?
I can't see it.
Oh, you can't see it.
Mm-mm.
All right, how about now?
Okay, yeah.
Okay.
in a relaxed muscle, actin and myosin myofilaments lie side by side, and the H zones and I band are at maximum width. During contraction, the actin and myosin myofilaments interact. The actins slide toward the center of each myosin myofilament. As a result, the sarcomeres shorten. In the fully contracted muscle, the ends of the actin myofilaments overlap, the H zones disappear, and the I band becomes very narrow.
Now, one thing to note here, as this sliding filament theory, works, does the actin or the myosin change overall length? Do either of them individually shorten? The answer is no. What changes is the interaction of the two. As the actin gets pulled toward the center of the sarcomere, it causes the Z lines to get closer together. But the individual actin or the individual myosin, shown here in purple and in green, they're not changing their overall length.
As they contract, are they overlapped more?
Sure. Well,
Yeah.
yeah, I mean, you can see the actin's getting pulled toward the center.
Right, okay.
But the individual myosin, the length here in the fully contracted state is no different than it was in the fully relaxed state. Here's full relaxation right here. The green is finite in terms of its length. When we get done at the end, it hasn't changed its overall length. It's still the same length. And the actins likewise haven't changed their individual lengths. What has changed is how they've interacted with one another relative to one another. So again, you need to watch these videos, and this will hopefully make more sense. This is the first time you've heard this. Now, what do you see on your screen now? You see what causes a muscle to relax, or you see the video?
What
causes
causes the muscle to relax?
Okay. All right. So we've just talked about contraction in skeletal muscle. Well, your muscles aren't in a constant state of contraction, are they? They have to go back to the relaxed state at some point in time. Otherwise, you'd be like fully tensed all the time. So think about what caused contraction. What initiated contraction? Well, you got to go back to the neuromuscular junction and understand that it was a nerve impulse that caused the release of the neurotransmitter acetylcholine across the cleft. that in turn initiated a muscle impulse. So it's absolutely critical you understand neuromuscular junction and what that involves. So in order to talk about what causes relaxation of the muscle, you've got to go back and you've got to get rid of the neurotransmitter at the neuromuscular junction. And so that's what the right-hand part of this table, 9.1, talks about. There's an enzyme called acetylcholinesterase, which, you know, it's an enzyme because it ends in ASE. And what acetylcholinesterase does is it breaks down acetylcholine. If you break down acetylcholine, can you generate a muscle impulse? Answer is no. If you can't generate a muscle impulse, will the sarcoplasmic reticulum continue to release calcium? The answer is no. If there's not a steady calcium supply on the troponin, which causes the tropomyosin to move, opening the binding sites, right? This all has to work in order. You're not going to get interaction. So what ends up happening here is you get rid of the acetylcholine, the calcium ions get actively transported back into the sarcoplasmic reticulum because there's no muscle impulse going on, causing it to be released. So the calcium gets pumped back in. That active transport process takes energy. Remember this from earlier chapter? Pumping against a concentration gradient causes the cell to expend energy. It's ATP usage. Do you remember us talking about active transport? Yeah, we talked about it. So active transport kicks in. Calcium gets pumped back into the SR, sarcoplasmic reticulum. If you remove the calcium from the troponin, the tropomyosin then blocks the binding sites of the myosin. Can the cross bridges come up and grab on if the binding site's blocked? The answer is no. You won't get relaxed. You won't get any contraction. The muscle will go back to its fully relaxed state. And so you're going basically from here upward now during relaxation. And that's because you've removed the calcium from the troponin by actively pumping it back into the SR. Because you got rid of the acetylcholine back at the neuromuscular junction. So I think the best thing for you to do is to really definitely get into chapter 9 in the next couple days, so that it's giving you some time to think about this, because we're into respiratory system next week already, you know. How about questions?
So I might be getting ahead of myself, but I just remember this so well. Like muscle tetany, is that later maybe? Okay.
Yeah, yes, that's just discussed.
Like that's like complete
Yeah.
contraction.
We talk
Is
about
that more to do with like lactic acid?
Yes, that's described and talked about in the PowerPoint.
Okay.
Yep, in the Zoom lecture. Yep.
Okay.
Yep. That's actually in section 9.3 under muscular responses. I talk about tetany, summation, recruitment. Yep. And then we wrap up the chapter also talking about the other two types of muscle tissue, right? We've been focusing on skeletal muscle, but there's also smooth muscle and there's cardiac muscle. Now, we don't spend a ton of time on those other two. We talk a little bit about them, but basically, when you get to the end of the cardiac muscle section, which is section 9.5, then from 9.6 to the end, that's pretty much muscle identification, which of course we're doing in lab. We started that today. We'll finish that up next Wednesday. So the chapter itself, in all actuality, if I count the number of pages, it's, let's see, 293 to 309. So what is that? 293, that's 16 pages. That's not really horrible, but there's a lot of information packed in those 16 pages. So just take your time, go through the PowerPoints, watch the Zoom lecture, watch the videos, watch the animations, and then come with questions next Tuesday. Because, really, we should be talking about respiratory system next Tuesday, but, we'll talk about whatever you want to talk about. But we still have to stick to the schedule. Which means no rest for you. Easter Sunday, nope. No Easter baskets, no chocolate, no marshmallow bunnies. Nope. You get into your A&P book. Study, study, study. Sadie's like, no way, not going to happen.
No, I already have a chem test that day and a pre-cal quiz and 2 chem labs to write up. So Sunday's booked for me already.
You have a test on Easter?
Yep, and a quiz as well.
This is Corey Damon? I'm going to talk
It's
to him.
like Thursday, it opens Thursday, and then it's open till Monday. But Thursday, like, we don't have our notes done yet, and then we have a homework on Friday for it. And then, like, over the weekend, I was just gonna do it. I don't want to wait till like Monday.
Yeah.
So.
No, I mean, Easter, you got to have some fun Easter. Do your parents, or do you hide baskets for your kids? Was that a tradition in your family?
We do like a scavenger hunt.
Mm-hmm.
I wonder if you go to other countries, whether they do that too. I don't really know.
I heard a joke once. It was like, what do we do on Easter? We have a bunny and he brings eggs. Why does he bring eggs? I don't know. What should we do with them? Hide them. Because it doesn't really, I don't know.
Yeah. Well, I went out to the store before I came to class today, specifically looking for marshmallow eggs. And I'm talking about the ones that are coated with like milk chocolate or dark chocolate, and then there's marshmallow inside, like a half egg. Do you know what I'm talking about? Not the Cadbury cream eggs, but the marshmallow inside. When I was a kid, that was like my favorite Easter candy, and I always had like 8 of them in my basket. I cannot find them anywhere. Maybe they're not as popular as I think they should be. I don't know. But,
Okay, for the answer sheet, I can come up with those.
No, you couldn't, unless you special order them, because I've looked.
There's a couple things online that make a bunch of stuff like that. I know there's like Milk something does a lot of stuff that I haven't seen in forever, and I can't remember what the other one is. They make it all and send it right to you.
Well, sure, yeah, you can get whatever you want, I'm sure, if you look hard enough. But yeah. Okay, well, I hope you have a good break, and do spend some time enjoying Easter with your family and all that. And get into chapter 9, and make that definitely a goal by the end of the weekend. If you can, try to get through chapter 9. That would be a really good thing to try to do.
So basically the second half of the chapter is just everything we're doing in lab, the muscles and stuff. Okay. So it looks like longer than it is.
Absolutely.
So like for the quiz, is it gonna be like more like muscles, or is it like, because we didn't really do much on joints?
Right.
Because there was like 2 questions, like I think on the practical today. So like I didn't know if the quiz was gonna have like anything of like the joints.
It's been so long since we've really had the joints on the schedule here, so I think we will focus on the muscular system. So let me give some thought to what sections in chapter 9. I'll narrow it down to a couple sections, not the entire 5 sections. That's just too much information, but let me think about that, and then I'll email you guys, like
Sunday.
no, before
Happy
then.
Easter.
I will make a note. I'll let you guys know like tomorrow or sometime. How's that? Okay.
Thank you.
Yeah, you're very welcome. All right. Talk to you later.
Take care.
Yeah, you too. Bye.
I sent her, I got a hold of her.
Oh, Beth
Yeah.
Lacey?
Yes.
Okay.
And I told her that you'll probably be contacting her, so. Or,
Oh, did you have a date picked out?
no, she said she'll just wait for you to send over the answer key, and then we'll go over it. We can make a date between Monday and Tuesday.
Okay, that's great. I'll send her the answer key right now. I'll do it right now while I think of it.
Okay.
And then you guys are gonna coordinate it on time. Okay.
Yeah. So let me get this right, and I'll let you go real quick. In this chapter, chapter 9.
Yep.
I'm starting from the beginning of the chapter, going to 309.
Yep.
I'm going so from the beginning to 309.
The top of 310 has a diagram you might want to look at.
Okay.
But basically, yes, that's right.
So know that by the time I show up Wednesday.
That would be ideal.
And then
Tuesday for review.
Tuesday for review. And
Yeah.
then the quiz is on everything from 311.
I'm gonna send you an email
Okay.
tomorrow. I have a note written to myself. I
All
got
right.
to give some thought to that. I will email the whole group and I'll say quiz number 4, lecture quiz 4, given next week in lab on the 7th, will be over, blah, blah, blah. And I will tell you what sections of chapter 9.
Okay.
Yeah.
All right. Sounds good.
Okay. Sounds great.
Hey, have a good
Thanks, John.
weekend.
Thank you. You too.
Bye.
Bye.